Nothisha abanethulela izifundo zika grade 12 zonke izinsuku kusukela ngoMsombuluko kuze kube yiSonto. Zithokozeleni lezi zifundo, nisithumelele imibuzo lapho nidinga khona usizo.
Sanibonani bafundi ba grade 12. Kusewuye uthisha Mabaso ofundisa life sciences ePhindukane Full Service High School. Saqhubeka na life sciences. In our previous lesson besikhuluma ngopaper 1 ama basic concepts in paper 1. Now we are moving on now to paper 2. And uma wena kusisa kulama previous recordings or previous lessons ekade ngiwenza bengidila ngopaper 2, that is genetics and evolution. Siselapho nje genetics and evolution are the very important chapters and they carry a lot of marks. For instance, genetics iyodwa is 35 marks, it's 40 marks. Bese kuthi evolution is 45 marks. Uma kuhla 66 marks. Uma kuhlanza kokubili kuba 110 marks. So uma ufunde kahle lama main chapters or key chapters, you are in a position to pass. All right? And also nakhona lapha futhi ke ngizokubalula ukuthi ke kumele uyazi terminology. All right? So nalana futhi I'd like you to have your pen and paper ready because uma ngiqeda ngale topic, so I'll have a session where we deal with a terminology in genetics and evolution. All right? And then let's look now at the structure of paper 1. So paper 2. Now paper 2, we've got very few topics. There is nucleic acids, protein synthesis, meiosis, genetics, as well as evolution. So they're it's a very small amount of topics as compared to paper 1. Okay? Laba paper 1 ke siqala nge-DNA, ama-nucleic acids. So ama-nucleic acids, we've got DNA kanye bana no RNA. There are two of them. DNA is deoxyribonucleic acid. All right? Why is it deoxyribonucleic acid now? Because it consists of the sugar, which is deoxyribose sugar. And then RNA yona is ribonucleic acids. All right? And these two kinds of nucleic acids are made by the structural units which are called nucleotides. They are called nucleotides. All right? Let's look at the biochemical structure of these nucleic acids. One, they have what we call the nitrogen bases. Nitrogen bases are adenine, thymine, guanine, cytosine, and uracil. So they are all 5 together. Then the first nucleic acid that we're going to talk about is DNA. DNA is a deoxyribose sugar. All right? So masibi is a deoxyribonucleic acid. All right? And let us look at the DNA structure. A DNA structure is double-stranded. It has got 2 strands. And these strands are formed by 4 different nucleotides. All right? And what is a nucleotide now? Nucleotides are structural units of nucleic acids. So like for instance, a nucleotide is comprises of the phosphate group or a phosphoric acid, a deoxyribose sugar, that is DNA, nitrogenous bases. Ama-nitrogen bases atholakala lapha ke ku-DNA. Adenine, thymine, guanine, and cytosine, our form. ATGC. All right? So each and every nucleotide is named after a nitrogenous base. Like for instance, we can have a phosphate, a deoxyribose sugar, and adenine. So that nucleotide is called adenine nucleotide. And please bear in mind, each and every nucleotide consists of these 3 things: phosphoric acid, or a phosphate group, the deoxyribose sugar, and the nitrogen bases. And these nitrogen bases in DNA, they are linked to one another by means of weak hydrogen bonds. All right? So with DNA, we have got 4 different nucleotides. That is adenine, thymine, guanine, and cytosine. And when it is double-stranded, so adenine always goes with thymine, and they're held together by a weak hydrogen bond. And guanine always pairs up with cytosine. All right? And uma sisibheka the structure of DNA, a phosphate group, deoxyribose sugar, let's say adenine. All right? So the adenine izohlangana ne-thymine or thymine, okay? And the deoxyribose sugar and the phosphate. And these 2 nucleotides are held together by the weak hydrogen bonds. All right? And the guanine always bond up with cytosine. All right? So it is double-stranded and it is highly coiled. That's why you call it the double helix. It is a double helix. So basically, the structure of a DNA consists of the phosphate group, deoxyribose sugar, nitrogen base, as well as the hydrogen bond, which forms a double strand. So it is like a twisted line, and that's why we say it is a double helix. All right? And this DNA is very long and highly coiled, and it carries genetic information or hereditary characteristics of individuals are contained in DNA. Yingakho ukuthi uma abantu bayotesta noma mhlawumbe ipaternity ukuthi ubaba ubani kwenziwa i-DNA. Uma thola umuntu efile engazi ukuthi wakuphi lo muntu loyo, kuthathwa i-DNA sample yenziwa i-DNA profile. And i-DNA profile is more reliable bakwethu because it is unique with each and every person, except for identical twins. But genotypically, we are different because our DNA are different. All right? So this is the main function of DNA. Okay? And the other function of DNA, it is responsible for the formation of proteins. And proteins, we know that they are the main building blocks of our bodies. All right? So, for this DNA to be carried from one cell to another, it must make the copies of itself, and that is called DNA replication. And so, meiosis, a phase interface where DNA replicates. All right? So, before we look at that one, let us look at what are the importances of DNA and why should this DNA replicate. One, it doubles to double the genetic material so it can be shared between resulting daughter cells during cell division. That is DNA replication, and their significance or importance. Two, DNA is responsible for the formation of identical cells during mitosis. So each cell must look exactly as the parent cell. All right, let's look at this mechanism of DNA replication. All right, step number 1, DNA or a double helix unwind. So njengoba sho that i-DNA ifana ne-steppiece esigoqekile esisongene. All right, so before it replicate iyaqhaqheka, iyavuleka, it unwinds. All right, and then remember between the nucleotides we've got hydrogen bonds. So the bonds are nucleotide ayanqamuka. Okay, they break into 2. Okay, and then once these bonds break, i-DNA ibisivuleka njengozip, it unzip. Okay, and separate into 2 single strands. And each of the original DNA strands serve as the template from which the complement is built. So remember, each strand yilapho kuselwa khona ukukhanda kwani kwenye i-DNA, because i-DNA fanele icopy efana nayo. So you should i-DNA 1 is able to produce DNA2 from each strand. Okay, and number strands which are formed, siwabiza ngama complementary strand. But let's strand lapho asiswela khona complementary strands thina a template. And you must remember, so if you are asked in an examination to explain DNA replication, DNA is used as a template. Template is very important. You must include that one either in an essay or in question 3 or question 2, where we are required to explain the process of replication. All right. Step number 4: the free nucleotides build up a DNA strand into each of the original strands. So lapha ku nucleoplasm kunama nucleotides aflotayo, okay, ayantanta nje all around. So mayivuleka le DNA la lama nucleotide ayidantelayo azehamba manje ayokhanda strand lesibili, that is the complementary strand. And lama nucleotides ke abesehlangana ke according to ilungana ye-DNA sequence. So le part ye template eno adenine ngapha kuzoba na uthymine. Uma kuyi-guanine kuza ucytosine. Uma kuyi-cytosine kuza bani u-guanine. Njalo njalo until and as it separates. Alright? Kukhandeka new strand. Alright? And then at the end, 2 identical DNA molecules. And each consists of 1 original strand and 1 new strand. This is the template and the complete. And that way, DNA is doing kabili. And that is DNA replication. All right. Let's look at now what is DNA used for. And it is called DNA profiling. DNA profiling. Sometimes we about DNA fingerprints. All right. So every person except identical twins has his or her own unique DNA profile. All right. And mobile DNA profile, it can be arranged as black bars representing DNA fragments of a person. And what are the uses of DNA? This is very important. We've got 4 main uses of DNA. 1, to identify the criminals. 2, to identify the dead bodies. 3, identify the relatives. And 4, for paternity test. So ngabamazi bathi la iDNA sayisebenzisa uma kade kwenzeke icala. Let's say kuhlasela umuntu mhlambe azagcine sefile. Kanti efatha bestrugglisha. Mhlambe lo muntu bexabana naye akhwebheke. Kusale nje very small piece lapha zolungana. So forensic experts ayakwazi to extract the DNA, all right, ye perpetrator, and compare it with DNA yalo muntu webehlasela. Okay? And then if ever bathola abantu belapho ku crime scene uzobathi uthaboke ngithathe DNA profile yakho, and DNA izosho exactly ukuthi ekabani. So like for instance, uma ushiya iDNA ku crime scene kufanele ukushiya igama lakho, isibongo, skin color sakho, height yakho, house number lapho uhlala khona, everything about you in a very small portion of DNA. Ngoba even uma sekuzima impela bathatha even toothbrush bayihwaye lezo nsana zama cell tissues mawucupha cell toothbrush, each cell, okay, can identify the criminals. So it is very used in identifying the criminals and crime scenes. All right, same thing makuthola lesidumbu somuntu ushonile akaziwa wakabani, kumele kuthathwe iDNA sample yakhe. Okay, emva kwalokho ke kuzosekuveza ukuthi ubani wakabani. All right, I thought zafika ihlobo zomfuno lapha thathwa iDNA samples bona ukuthi cha lo wakolo. So all right, and also to identify relatives ukuthi ngempela isihlobo sami yini wena maqhamuka usumzala nje, yabona, o aida uzala ubaba wami thina sikwazi. Just take the DNA yakho ne DNA kababa sithola ukuthi ngempela ubani wena. Lokho kuhambisana lo kuthiwa i-paternity test. Ukuthi ke usepregnant umama uyayithola ingane, hayi ubaba ngenekhanda, adaya kusiyona yami. Lokho ke siyakwazi through i-genetics angithi ukuthi ubaba heterozygous noma heterozygous, okay, for uba mnyama in complexion, bamnyama bobabili. Nangu mama siqhula izingane ezimhlophe, And total, let's look at two characteristics. Ubaba mnyama mude. No mama mnyama mude bayashada. Mase zala umama uzala ingane ezimhlophe ezishana. Eqala ubaba khala uthayiphe le ingane ayisifani nathi. Complexion yakhona, height yakhona kusiyona la ekhaya manje. Okay, kube sekuthathwa i-DNA ke yalabazali nama-DNA ey'ngane. Uma ama-DNA yabe matcha iy'ngane zakhe. Well, thina ke with the knowledge of genetics siyazi ukuthi siyahlehla, we look at the grandparents. Uma ngakubheda lapho futhi siyahlehla, we look at the relatives. So siyakhumbula ke i-pedigree diagram. It show ekhombisayo ngempela if you ukuthi sura lapha uzuyofika kuphi na kubaZulu. All right, so those are 4 importances of DNA profiling. All right, siqedile DNA. Then move on to RNA. RNA icishe ifane DNA. Umehluko wakhona ukuthi RNA is single-stranded. Okay, and okwesibili, instead of thymine, i-RNA has got bana uracil. All right, and also it is shorter than DNA, and it is mainly it can be found in the nucleus, and also can be found is mainly located in the cytoplasm. We've got a messenger RNA which is synthesized inside the nucleus, ikhandela phakathi nucleus bese iyaphuma iya ku cytoplasm iyosiza ekukhandeni ama proteins lo sibiza ukuthi na i-protein synthesis. So for examination ofanele ukwazi ingavela i-question ethi tabulate the differences between RNA and DNA. Once you see the word tabulate engqondweni akufanele khala ukuthi vele usunalo imali elani makla lokudweba i-table. All right, and we divide it into 2. Ufaka DNA ngapha ufaka na RNA. So these are marks to ama free floating marks noma ama low hanging marks. Okufanele umfundi life science angela ama marks afika wathola. Makubuza nje umbuzo kanjena nje o tabulate o graph. All right, so abone nje ama marks as time goes on. All right, akasibheka ama differences. One, DNA is double stranded. So RNA is single-stranded. So lena ingabili stepis kanti lena ihamba nga1. Lena inoshukela kuthiwa i-deoxyribose sugar. That's why we say DNA, deoxyribose sugar. Le ngapha ine-ribose sugar. That's why we say RNA. And thirdly, it contains a nitrogenous base, thymine ngapha khona ina uracil. And those are the differences between DNA and RNA. Sifundelani na i-DNA na RNA na. So lesizokwazi ukubona ukuthi ama proteins akhandeka kanjani. I-RNA ne-DNA are responsible for the formation of proteins by the process which is called protein synthesis, which takes place in every cell of living organisms. So these 2 topics, they link to one another. Okay, DNA amanyucleic acids kayilana ne protein synthesis. That's why but here DNA is the code of life. All right, akasi define a protein synthesis lapho. Protein synthesis is the process by which proteins are made in each cell of an organism to form enzymes, hormones, and new structures for their cells. So the function of proteins, they are the main building blocks of our bodies. Like for instance, we have got enzymes which control the metabolic reactions in our bodies. We have got hormones as endocrine system lab, and they bring about coordination, chemical coordination. And they are proteins in nature. Although proteins are kind of various structures in our bodies, that's why the main function of the body, it is the main building component of living cells, both plant and animal cells. All right. So, low stability protein synthesis. All right. And there are 2 main processes in protein synthesis. That is transcription and translation. All right, let's look at the mechanism of protein synthesis now. We'll start with transcription. I-transcription is a process of the formation of the messenger RNA, and it takes place only in the nucleus of the cell. Alright, lapha ngifuna ukuthi ubheke ma-senza i-protein synthesis icishe ifane eqaleni na ne-DNA replication, because nakhona lapha DNA unwinds and split into 2 strands. Alright, and each of the strands act as a template forming the messenger RNA. Okwesithathu siyafana ukuthi 3 nucleotides arranged to form messenger RNA according to the template, and this is called transcription. Alright, then what happens now? Messenger RNA then leaves the nucleus through the nuclear pores, okay, to the cytoplasm, where it becomes attached to the ribosomes. Those are translocation. So uyabona lapha ukuthi ke kuqale kukhandwe i-messenger RNA. Igama lazo messenger, it carries a message from the nucleus. Nucleus ihambe nawo iphume through i-translocation iye ku-cytoplasm of the cell. Mayifika lapha ku-cytoplasm isiyasho ukuthi kufanele kukhandwe hlobo luni lani lama-proteins, and masifikisho kanjalo ke sesithi na u-translation, to translate. Okay, njengoba njengoba ngifundisa manje fingisho into ngesingisi i-translate ngise kuZulu angithi ngithathe from isiZulu and I translate it ngise kuphina ku-English so that you can better understand what I'm saying. Same thing nalana, le message lena which comes from the nucleus iphethwe uba na messenger RNA, alright, is carried to the cytoplasm and it becomes attached around the ribosomes. I-message itholakala ku-genetic code lo kuthiwa i-codon. Uma isikhandekile mRNA, ama-nucleotides ahamba ngamathathu. These 3 nucleotides are called the codon or the genetic code. Like what I said to adenine, cytosine, guanine. That's a genetic code. And remember, ngikhathi khangela about DNA, lapho kade khona adenine. Akube kusaba u-thymine ngayodwa, but instead uba na u-uracil angithi. So that u-thymine is replaced by uracil during the formation of the RNA. So which means that the messenger RNA is synthesized from DNA. So then, once this code is inserted around the ribosomes are the cell structure which are responsible for the formation of photosynthesis. Actually, indawo lapho kukhandeka khona ini ama-proteins. Alright? So then once this connected genetic code isifakiwe lapha, sekusuka enye RNA manje kuthiwa i-transfer RNA esiyibhala to tRNA using small letter t. Lapha ku-messenger RNA i-mRNA we use small letter m. And these transfer RNA reads the code, all right, and interpret it. And that interpretation will cause amino acid to bring a specific amino acid for the synthesis of a specific protein. And this is called translation. And these amino acids are linked together to form a protein chain, all right, which is called a polypeptide. It is called the polypeptide. And each and every amino acid is linked to one another by means of the bonds which are called peptide bonds. Peptide bonds. Lana mahlangana ama-amino acid ke uma kuhlangana awu-2 sithi dipeptide. Okay? Mahlangana i-dipeptide nenye amino acid sewu-3 sewuyina a tripeptide. Okay? Once about more than 3 sesiwabiza ukuthi ama-peptones. Alright? Mase maningi so noma sokuthina ama-polypeptides. But once these amino acids ahlangana aba above 50 and above, then we call them the protein. So if amino acids are less than 50, we call it a polypeptide. Poly means many peptide bonds. But at 50 and above, then it is a protein. Alright? And lapha we have got 20 natural amino acids. The amino acids they are linked together according to the code or the message that has been brought by DNA velocity codon. Uma transfer RNA yofunda codon yona ifika na ne-anticodon. Alright? Now for instance, when DNA triplet yakho beku GAA, angithi kuzona masikhandeka mRNA kuyoba na uCUU. Uyahamba lo CUU uyongena lapha kuba na ku ribosome. Bese yaqhamuka transfer RNA imfunde. Lo CUU uzoba na uGAA. Guanine goes with cytosine. Adenine goes with uracil. Best by amino acid tuba na leucine. Alright? Ebafundu ningazixaka ukuthi nizo nafunde noma creme amino acids. Whenever they ask you questions concerning amino acids, they're going to give you the names of the amino acids. Ufana ukwazi nje ukuthi phela wona there are 20 natural amino acids that are found in living organisms. Alright? Siphela kanjalo ke ku my nucleic acids, that is RNA and DNA. Alright? Then the next chapter, paper 2, is meiosis. 12 marks. So I'm going to clear meiosis because we've discussed it in paper 1, econ paper 1 and paper 2. Alright? So when you find out phases of meiosis, and also with lapha during interphase, replication of DNA. Why now? DNA to make sure that the daughter cells have got the same genetic information as the mother cells. Alright? And also for doubling up of the genetic information before the beginning of any cell division, either mitosis or meiosis. Alright? Also, meiosis is linked with all the other chapters. Human reproduction is genetics, meiosis, and even evolution, meiosis. So meiosis is very important. Chapter 5 and it's found in both papers. And like paper 2, 12 marks. Okay? And also remember, meiosis also bring about a variation. And our cause of variation, difference is free floating marks. You crossing over during prophase 1. ASB, random arrangement of genes. Alright? During metaphase. AS3, ubana mutation. Non-disjunction. AS4 is chance fertilization. With any sperm can fertilize any ovule. So I go to here, Zalabafana pair, no Mazalamatoma, Pelamfana Tomazan. I would have 2 boys and 2 girls. Those would here a chance fertilization. So any sperm has got a chance to fertilize any egg or the ovule. And those are the causes of variation. Lapo ilo nganyama ye meiosis ofana wikhumbula. Then the next chapter I see discuss is genetics. Alright? We have discussed other aspects of genetics. So, genetics in the following way. 1. You must know the experiments performed by Greco Mendel, and you must be able to explain the 3 Mendelian principles. Principle of dominance, alright? Principle of segregation, and the principle of random assortment. So, just to give you a clue or to summarize this, the principle of dominance, one phenotype of only one parent is displayed. That was not dominant gene, no recessive gene. It means that between these 2 genes, genes, they always occur in pairs. Those alleles. Okay? So this is a revision from its DNA, its genetics. Alright? As far as genetics is concerned. 2. Crossing. Monohybrid crossing. What was during monohybrid crossing? Okay? Where we look at only one characteristic. Alright? And the cell lab during monohybrid crossing, we also look at the different types of dominance. Complete dominance, lapho a new characteristic is formed. Ingane ifana nabazali, flower elibomvu nelimhlophe mahlangana khipha ingane pink. Core dominance, both characteristics are displayed on an offspring. Iflower elibomvu nelimhlophe bazali ingane lamaqaba amhlophe nabomvu. All right? About complete dominance, it does matter ukuthi the offspring display the characteristic of only one parent. All right? Esinjwe lapho okunye kufanele ukwazi ku-genetics is the dihybrid crossing. What kwenzakalani during dihybrid crosses, and you must be able to solve ama-genetic problem. Selabo lo-genetic crossing nanka amanye ama-marks, low hanging marks. Uma unikezwa i-genetic problem, they give you the phenotype of the individuals, and also they state whether those individuals are heterozygous or homozygous. Thirdly, a question, gene dominant. Do you understand? Those are the three main things. Often as mouse to a panel diagram or genetic diagram, or you represent about 6 marks. So this is the format. I'm a max la umuntu angene mawuthi ulalela njengamanje usuna 4 over 150. Kanjani you write first parents P1 and their phenotype. All right? A tall tree is crossed with a short tree. Kushiwo lokho imakulelo free mark yabona na. And ngenzansi abaza ukuthi is either homozygous or heterozygous. Uma a homozygous azofana ini ama alleles. Uma a heterozygous azohluka ama alleles. That is the second mark. Okay? Free floating now, low hanging mark. And then lapho singaqaleka nalo mawukhipha ini ama gametes. Kodwa wazi ukuthi ke makhishwa ama gametes enzeka by which process? Meiosis. Uyambhala umeiosis, okay? Emva lokwenzeka ufertilization. All right? During fertilization, there's a male gamete fuse with a female gamete. And fuse but all the nuclei of the male and the female come together. We call that fertilization. So as you want that my favorite stage is fusion. Fusion and you write fertilization. And the master process is putting we call it the first filial generation. F1. That's why I keep that. Alright? So stay marukala, phenotype, LSB genotype. P1, no F1 it gives a mark. It started a mark. Meiosis, no fertilization, it gives you another mark. So out of 6 already we've got 4 marks. Not a statement only as well so as a genetic problem. Okay? So it's lobby from Ubalula genetics. And Koshua futi or selective breeding, genetic engineering, or cloning, as well as stem cell research. And lastly, ukumbule nkosiyami ucrossa. When you cross sex linked characteristics. Uma kuyifika uqala ukubhala ngo-XX bese kuthi leyo characteristic leyo like hemophilia or color blindness uyibhale phezulu. Sizwa lapho wethu. Please don't forget that one. And also before your final exam you must study these following genetic disorders: Down syndrome, color blindness, albinism, yes, and hemophilia. So bazokubuza lokho. And also you must be able to sex determination. No XX, no X, Y. What is it, a boy or a girl? And don't forget, crossings, genotype, using a phenotype, you give the genotypic ratio as well as the phenotypic ratio. Alright? And then the last subtopic in paper 2 is evolution. And evolution, remember, it is 66 marks. So most of the terminology is coming on. And evolution is we've got general evolution as well as human evolution. So general evolution you must be able to define the term evolution, change of organisms over time. And then also best evidence of evolution the organisms that we see today or that are existing now originate from organisms that originate from the past. So lapho uzokhumbula oma fossils angithi comparative embryology angithi ke homologous structures, okay, okusilinkayo nama ancestors. Then bese ba nama early theories amabili uDarwin nobani noLamarck. Uthini uLamarck na? Uthi uLamarck any part of the body noma yiphi yomzimba oyisebenzisa kakhulu iyakhula ibe nkulu ibe strong. Leyo ungayisebenzisi iyancipha and eventually disappears. Bese uthi le ekhulile ngathi ke usurvivor uma usuzala usuyidlulisela enganeni zakho. Okay? And there is the principle of inheritance of acquired characteristics. Ungamkhohlwa uLamarck uthi na law of use and disuse abathi na acquired in inheritance are passed on to the offsprings. But tell me about science by treat. But no, I Lamarck ngeke. Organisms do not change because they want to change. Obviously, there is no evidence. Ukuthi ngempela lama characteristics owathola amawukhulu. La for instance uthi ufuna ubane efube enkulu. Ujima sibona isikhosha ngefube engaka. Uthi masuzala ke uzala igona zona. So nazi lapha yaphetha khona itheory kaLamarck. So fanele ubhekisise and kuyazimlation mayibuza ngoLamarck they give you the examples. So ungavele uchaze as long as it is njengoba uyifundile. So mayikhuluma ama giraffe nakhuluma ngama giraffes. Mayikhuluma ngama cacti plants kuleyo lokungala yakho ukhuluma ngama cacti plants, okay? Eyesibili theory ilega Charles Darwin okuyona esasebenza kwamanje namanje. All right? Yana natural selection. Nayo siyenzile kakhulu in our previous lessons, so ngeke ngisagcina kangako. Ufanele ukukhumbule nakho nje ukuthi isequence ithini. 1. Organisms produce a large number of offsprings. These offsprings show a great deal of variation, and the line variation theory. All right. In these variations, some have got favorable characteristics and some have no favorable characteristics. So, they undergo a competition for the resources which are limited. All right. They compete for limited resources like food, like water, like shelter, and many other needs for living organisms from their environment. So la competition. And then during this competition, those with favorable characteristics are going to survive, and those with unfavorable characteristics are going to decrease or decline in number and eventually disappear and become extinct. Which means that only those with favorable characteristics will be able to reproduce, and the whole population is going to change. You know, a survival of the fittest. So this is just the reminder general evolution. Then we move on to population evolution. Lapho we are tracing imvelaphi yethu as human beings. All right? And we're using yona lendlela kabana kaLamarck lendlela kabana not kaLamarck kaDarwin yana natural selection umashintsha over time. And la natural selection ubukhumbuthi uDarwin benoLamarck bathi yenzeka kancane over a long period of time nama changes amancane. That's why kunama gaps in between the fossils. All right? And of late, scientists are come on the punctuated equilibrium. But about now, organisms they do not change for quite a long time. And that long time is punctuated, those punctuation marks, stopped, full stop. Okay? By short, rapid changes. Okay? And these changes take place faster in a very short space of time. And that is called punctuated equilibrium. All right? So okay? And thina ke ukuze sikwazi ukubona kahle umehluko phakathi kwethu noma development yethu, we compare ourselves with the primates. We compare ourselves with other primates because we follow the same group of primates. Mankhuluma primates khuluma ngabo African ape. All right? Of which uma kubhekwa ama fossils ethu ne evidence yakhona, we have got structures which are similar. Low shutina, it shows that we develop from a common ancestor. All right, but we are all primates. Then, so we are similar to other primates, and also we are different to other primates. So now similarities between humans, or Homo sapiens, and other primates. It's fun, man. Just look at yourself: the gorilla, the chimpanzee, the monkey, which is fine. So our eyes are in front. We've got the skull, which in the house is there, praying. We've got a freely rotating arm. We've got the long upper arm, which is a humerus, and the lower arm, which is freely rotating. And we have also the opposable thumb, which brings about power creep. It's not primates precision. We use power creep. So this kati to an opposable thumb because the thumb is always opposite to other fingers. We have got flat fingernails. Those are all the similarities between humans and other primates. Same thing with the differences. No matter what, because we've done it. Consider a lesson in evolution. But come out here, see, we've got a larger brain, they've got a smaller brain. Okay? And we've got a less protruding jaw, they've got a more protruding jaw. Which are prognathus, and more prognathus, less prognathus. Okay? So now on our own, after this lesson, we are tika noma you are writing down njengoba a pen nepaper. You are writing down izinto oqala kahle abuzwa ngami noma ubona ukuthi awuzazi kahle, and you go and study them. You still have time. You still have got the whole week. Okay? So for the whole of next week, we will funda life sciences because we on Friday as well as on Monday. So lana mean I'm giving you the overall view what yini ofanele uyifunde for examination. Manje inhloso yethu is to collect the marks. And please, cela unakisize ukuthi ngilongo wamenshina ama marks amahala. Mshina table. Ngamenshina abantu manje awe monohybrid crossing angithi amanye ama marks wathola ku graph. Ngokubhala nje heading. But the X and Y axis where we label kahle mama imark heading imark correct type of graph elinye imagla ama marks la owathatha ungaqala ukuthi ucabange i-answer to the question ungakabuzwa lutho usafunda nje question. That's why it is very important to read the question carefully before you answer the question. And abafundi banokuthi ke baphendule into right endaweni wrong wrong endaweni right inxa uthi uyajaha. Umuntu nje ezothiwa khuluma accommodation asabe ukukhuluma ngani uyazi accommodation usho near object or far object kanti nje bekhuluma ngo near object noma ngoba nango far object. Kwesikhathi baphambisa futhi pupillary mechanism lapha ehlweni noba na accommodation. All right? And then also enye into ofana uyazi as umfundi ukulandana kwethu nokhokho bethu kusukela ku-Ardipithecus, Australopithecus afarensis, Australopithecus africanus, Australopithecus sediba, Homo habilis, Homo erectus, and Homo sapiens. Lapho ucheka age, and also ucheka ukuthi yatholakala kuyiphi indawo, and also ucheka ukuthi yathola ubani, and look at the different characteristics of these different stages or different phases of development of humans from African apes up until Homo sapiens, Homo sapiens, and we are much wiser than any other organisms. All right, and then lastly, we've got this one which is called the cladogram or phylogenetic diagram. So please study this phylogenetic diagram. How do you branch from our main ancestors? No Homo nantalis. Just really true. Ikona lapho mind the gap ikona kuzonke imali the pedigree diagram or the cladogram. Sengivala ku paper 2 human evolution out of Africa hypothesis. lena ngizophinda futhi ngithi uyigcizelele phela out of Africa hypothesis. All right, ushiye ngani ukuthi out of Africa hypothesis because kuthiwa bonke abantu abakhona emhlabeni badabuka la eAfrica. Aba China, ube German, ube English, ube India, but imvelaphi yakho is from here eAfrica. Abaphika bathi mina ngifani neChina, mina ngifani neIndia. Yabona? Yebo, uqinisile. Remember, when organisms increase in number, they separate through speciation. Angithi? Kwenzeke allopatric speciation. Each individual, there's no gene flow, and each individual develop independently in each environment. Kodwa sonke njengoba silamhlabeni sisuka kuphi na la eAfrica. Nansi evidence noma nansi izasho kazi na. One, the oldest fossil is found here in Africa. Magumbiwa ama fossils endala nawo wonke ila eAfrica. We have got the evidence. But qhubeka nokuhamba namanje, okay? Uma mhlawumbe kwenzeka ukuthi sithola i-fossil endala mhlawumbe eIndonesia, then kuyoshintsha ke ukuthi no, they need to be out of Indonesia hypothesis. But okwamanje, you must know that all human beings originate from, and evidence provided through ama fossils, okay? Ama fossils endala nawo wonke fossil ila eAfrica. Also kubhekwe futhi ne genetic evidence. Nebona siyabuya manje ke genetics kanye lana ne DNA. If you look at the DNA gamma, siyakwazi ukutrace i-mutation ihlehle iziyokhomba umama uqala owayiqhamuka dabo khona la eAfrica. Abanye abantu manje why i-DNA kamama la siyahlehla futhi sibuyela paper 1. During fertilization, uma i-sperm sipenetrator noma singena eqandeni sishiya ngaphandle i-DNA, which means that i-DNA kababa ayingeni enganeni. So i-DNA ingane khona njengoba nisibona nje la ama-DNA siwaphethe ama-DNA akaba na akamama. All right, so kuyazeka ukutrace ke i-DNA yama-female sikwazi ukuthola umama uqala. Enye genetic evidence futhi ilapha kubaba X and Y. We know that umama no XX, ubaba no X and Y. So abantu besisa kuphela abantu Y. So siyakwazi ukutrace sibuke i-mutation of i-Y chromosome until sifike present. Kuyabona nakhona futhi indoda yokuqala eqhamuka khona lapha kwelakithi eAfrica. And kungakho ku paper 2 bantu bami. Kungakho nje ku paper 2. And please go and study. Go and study. Particularly ama differences. Ngakavali nje ama differences. Meiosis 1 and meiosis 2. Natural selection, artificial selection. Darwin, Lamarck. Mitosis, meiosis. Okay? So nje fanele ungene exam uwazi wonke. And mark 2 calculate, don't forget to get that free mark. To draw the table. And I wish you good luck in your examination. Nipass lab sciences. And we must have the better future. Bye bye. Good luck.
Siyanibonga ngokufunda nathi. Siyanimema ukuthi nithamele futhi nakusasa. Ningazithola lezi zifundo ekhasini lethu lika Facebook elithi Go Digital SA Foundation na ku YouTube elithi Go Digital SA Foundation. Ziyatholakala na kuKhozi FM njalo ngoLwesine kusukela ngo9 kuya ku10 ebusuku. Ningasithola nango WhatsApp kwinamba yethu ethi 068 151 1072.